Hello. Today we are continuing our conversation on neurons, and we'll be focusing on the synapse. Our learning outcomes for this section include defining synapses, identifying the components of the synapse, and explaining the events that occur at the synapse. So let's begin with the synapse. What is it? So our synapses are intracellular junctions that exist between excitable cells. So between neurons, or between neurons and a muscle, or even between different muscles. More specifically, synapse means to clasp or to join. It is a junction that mediates information transfer from one excitable cell, such as a neuron or muscle, to another. In our bodies, we have two main types of synapses. We have electrical synapses and chemical synapses. So let's look at these. Here we have an image of an electrical synapse and of a chemical synapse. So our electrical synapses are composed of gap junctions. You may remember gap junctions from earlier conversations. Gap junctions are a type of intercellular junction that's fluid-filled. They're fluid-filled channels through which ions pass from one cell over to another cell. So they're directly connected through these gap junctions, through these connexons. Now, electrical synapses permit the direct and rapid communication of impulses from one cell to another. Electrical synapses are common in our smooth muscle and in our cardiac muscle. Chemical synapses, on the other hand, occur between cells that are not physically connected by, say, gap junctions. So here we see one cell. Here we see our second cell, and there's space between. There's not that direct connection. So instead, these cells rely on chemicals called neurotransmitters, and these chemicals, neurotransmitters, act as a go-between in the space that separates the cells. Chemical synapses are common between neurons and between neurons and an effector tissue, such as a muscle or a gland. While it is important to recognize both types of synapses and to recognize the differences between these two synapse types, we will be spending the majority of our time talking about chemical synapses. So more on those now. Chemical synapses have a few key components. There is a presynaptic neuron, which is in front of the synapse. The presynaptic terminal releases neurotransmitters from the presynaptic vesicles in the axon terminal, so in these very end parts of the axon. A synapse also exists in our chemical synapses, and these are the spaces between. So our synapses are places of chemical communication where the neurotransmitter that was released from the presynaptic terminal diffuses from one cell to the next through the intracellular fluid. And we have a postsynaptic neuron or effector cell which receives the chemical signal. Now, our presynaptic and postsynaptic neurons are like two little girls playing catch. The presynaptic neuron is the little girl who throws the neurotransmitter, or the ball, neurotransmitter in our neuron's case, to the postsynaptic neuron, little girl. And the postsynaptic neuron is the recipient of that neurotransmitter. Now, the terms presynaptic and postsynaptic are really reference terms. They're used when discussing the communication between two neurons. For instance, the neuron that was once considered a postsynaptic neuron can become a presynaptic neuron when we talk about its communication with another neuron, if it then passes a neurotransmitter onto the next neuron in line. Chemical synapses take place most commonly between the axons of the presynaptic neuron and the dendrites of the postsynaptic neuron. This type of chemical synapse is called an axodendritic synapse, where the axon of the presynaptic neuron is talking to the dendrites of the postsynaptic neuron. We'll spend the majority of our time talking about these. Sometimes, though, the axon of the presynaptic neuron forms a synapse with the soma of the postsynaptic neuron. This type of chemical synapse is called an axosomatic synapse between the axon of the presynaptic neuron and the soma of the postsynaptic neuron. Additionally, we can have a third type of chemical synapse where the axon of the presynaptic neuron forms a synapse with the axon of the postsynaptic neuron. This is called an axo-axonic synapse. While we will keep our discussion of synapses pretty straightforward, do keep in mind that one neuron can have 1,000 to 10,000 different axon terminals and can be stimulated by 1,000 to roughly 10,000 different neurons. So, needless to say, our neuronal networks are crazy complex and incredibly sophisticated. So, whenever many neurons, such as those shown here, form synapses with one neuron, we say that the pathway converges. On the other hand, when one neuron branches out at its axon and interacts, forms synapses with many neurons, and then those neurons form synapses with many more neurons, we say that the pathway diverges. All right, so let's take a closer look at our synapse. Here we have a presynaptic neuron. This neuron generates an action potential originating at its axon hillock and conducts that action potential down the axon toward the axon terminal, shown here. Here we have our chemical synapse, this junction that mediates information transfer from one neuron to another using neurotransmitters. And here we have our postsynaptic neuron, which receives the neurotransmitter and converts it into an electrical signal, which gets transmitted away from the synapse toward the cell body, toward the axon hillock. Looking at an electron micrograph of a chemical synapse, we see the axon terminal of the presynaptic neuron here. The synapse, the space between, or also called the synaptic cleft, and the plasma membrane of our postsynaptic neuron. Inside of the presynaptic terminal, we have many mitochondria, which indicates high energetic demands, and we have several synaptic vesicles filled with neurotransmitters. We know that the neurotransmitters ultimately get released and get passed from the presynaptic terminal to the postsynaptic neuron through that synaptic space. But how does that happen? Well, let's take a closer look. So here again, we have our presynaptic neuron. So our presynaptic neuron fires an action potential, and that action potential travels down the axon to the axon terminal. Now, the axon terminal does not have voltage-gated sodium or voltage-gated potassium channels. Instead, the axon terminal has voltage-gated calcium channels, and these channels open in response to the action potential. And whenever these channels open, calcium, which is high outside the cell and low inside the cell, flows into the cell, into the axon terminal. This calcium entry ultimately causes the synaptic terminals filled with neurotransmitters to exocytose and to send their neurotransmitters into the synaptic space. Now, the neurotransmitter in turn diffuses across the synaptic cleft, this space between the cells and binds to specific receptors on the postsynaptic membrane. This binding then causes the postsynaptic neuron to respond, often by opening ligand-gated channels, allowing certain ions to flow through, and causing graded potentials in the postsynaptic cell. Now, it's not beneficial for neurotransmitters to stick around forever. So that would create prolonged and unnecessary activation of the postsynaptic neuron. So we want to clear the slate. We want to get the neurotransmitters out of there. So there are three main ways that this happens. One, the neurotransmitter can be enzymatically degraded. It can be broken down by enzymes that are either in the plasma membrane or in the synaptic space. Neurotransmitters can also be taken back up into the presynaptic terminal through reuptake transporters, and then those neurotransmitters can be recycled and reused. And sometimes the neurotransmitters just diffuse away from the synapse, and so no longer have an effect on that postsynaptic neuron. Now, let's add just a tad bit more information to what is happening in our presynaptic neuron, specifically in the presynaptic terminal. So, as we know, an action potential originates up in our axon hillock. It travels down the axon to the axon terminal and causes our voltage-gated calcium channels to open. And calcium, consequently, enters into our axon terminal through these voltage-gated calcium channels. Now, whenever calcium enters in, it binds to a sensor protein. And this sensor protein is called synaptotagmin. Synaptotagmin. It's a sensor protein. And so calcium and synaptotagmin form a complex. And together, they travel down to the end part of our axon terminal, ultimately to interact with our vesicles. Now, the synaptic vesicles, here we see a synaptic vesicle filled with neurotransmitter. These are being anchored in place by something called snare complex. So a snare complex is just a complex, a grouping of proteins that get the name snare. And these help, again, anchor our vesicles in place. But whenever calcium and synaptotagmin travel through the axon terminal down to the snare complex, they cause the docking and the fusion of those synaptic vesicles with the plasma membrane. So here we see the complete fusion of this synaptic vessel, which is ironically made of a phospholipid bilayer, and it fuses with the phospholipid bilayer of our plasma membrane. And this causes the exocytosis of the neurotransmitters into the synaptic space. Again, those neurotransmitters travel across the synapse and bind to specific receptors on that postsynaptic membrane. But it all happens in response to an action potential being fired. That action potential causing calcium to enter into the cell, calcium binding with synaptotagmin, and then together traveling down and interacting with snare, with these proteins that anchor our vesicles and cause then the vesicles to dock, to fuse, and to exocytose. And again, at that point, the neurotransmitters will diffuse across the synapse and bind to specific receptors on the postsynaptic membrane. The effect that this has on the cell will be discussed in our next lecture. So at this point, we should now be able to define a synapse, to identify the components of a synapse, and to explain the events that happen at the synapse. So until next time, have a beautiful day.